Welcome to episode 2 of Sky Biology, friends. And in this episode, we are going to discuss about the ion channels across the membrane of neuron. Now, why are we discussing these ion channels across the membrane of neuron? Because, if we want to know about the nerve impulse transmission across the neuron, it is very necessary to understand, how this nerve impulse is passed along the neuron or conducted along the neuron. And whenever we are talking about nerve impulse transmission, we are basically implying the transmission across this axon. Now, as you know, we already discussed in the episode 1 that, what is this neuron all about. Let me just add on to it. When I said there is cytoplasm in the neuron, cell body of the neuron, of course there is cytoplasm in the axon also. When I said there are cell organelles over here, then of course it implies that there are cell organelles in the cytoplasm of the axon as well. Fine. And I also said, if you remember, then this only thing is that you have to remember for your NEET exams and that, Nissls granules, the rough endoplasmic reticulum masses are absent in axon. Okay, so let's take this further. If the neuron is myelinated, which in this case is myelinated, then even if these ion channels, which I have shown, one over here, are there across the cell membrane of the cell body and the axon, then the ion channels are actually mainly concentrated in the areas where there are nodes of Ranvier or where the myelin sheath is absent. Okay, and when I talk of nerve impulse transmission, when the neuron is actually myelinated, then the nerve impulse transmission is actually from one node of Ranvier to the next node of Ranvier, because due to insulation or myelination, the nerve impulse along those areas where myelin sheath is present doesn't die down easily. So the nerve impulse jumps from one node of Ranvier to the next node of Ranvier, because the movement of that particular impulse is so swift over here. It is as good as we say that it stops only at these areas to go further. Okay. So, let's talk about one such ion channel. Now, you may be wondering what is the structure all about. So, this is the membrane of axon that I have tried to zoom in over here. And, of course, the membrane of the entire cell is not as simple as it seems to be. It somewhat looks diagrammatically like this. One more thing before I proceed further. If there is a stimulation of the neuron, so then the stimulation is through the dendrites, because the input signals are received through the dendrites. I mean to say that the dendrites are sensory in function. So, whenever there is stimulation, the stimulation is received through the dendrites. And if the stimulation is high enough to cross the threshold stimulus, which is around minus 55 millivolts, then it is strong enough to conduct a nerve impulse. And if the stimulation is something which is below this, that is minus 55 millivolts, which is the threshold stimulus, then the nerve impulse is not generated within the neuron. So once again, there's an important concept or term over here, threshold stimulus. Threshold stimulus is the minimum intensity of stimulus which is required to create an action potential or a nerve impulse along the neuron. Okay, so let's zoom in on this cell membrane of the axon. The cell membrane of the axon is somewhat like this, or for that instance, let me wait and say the cell membrane of any cell for that matter, or the cell organelles. The cell membrane of the cell organelles also is somewhat like this. Now, why is it like that? Because anything that wants or that needs to come inside the cell or leave the cell has to be very selective. Anything cannot come within the cell or leave outside the cell. The cell membrane gives a very important property to every cell, that selectively that is selective permeability. So this is, let's stick to the neuron. So this is the cell membrane of the neuron, specifically the axon, which is a lipid bilayer. What do I mean by lipid bilayer? You can see there are two structures which are facing each other. Their tails are facing each other. The head is made up of phosphate. I have written over here phosphate head, and the tails are made up of fatty acid. Now, this is also known as phospholipid bilayer because of this. So now anything that has to pass doesn't find it easy to pass along this. But then there are certain protein channels or ion channels embedded inside the membrane of this neuron, so that if certain thing has to pass through it, that particular ion channel is selective or is very specific for that particular thing to get attached to that ion channel and pass across the membrane. Now first we are going to talk, there are 4 ion channels found in the membrane of neuron. One is sodium potassium pump, the second is passive ion channel, the third is voltage gated ion channel, and the fourth being ligand gated ion channel. We are first going to discuss sodium potassium pump. As the name suggests, it's a pump. That means it requires energy to do something, and the energy comes from ATP, which is adenosine triphosphate. It is therefore also known as sodium potassium ATPase pump. We can also call it sodium potassium pump for short. Okay, so this particular lipid bilayer is over here, and there is a protein ion channel or sodium potassium ion pump. Now also let me mention this, that in my diagrammatic representation it seems to be very simple. It's just a diagrammatic representation. The protein ion channels, since they are proteins, they are very complicated molecules, and the structure is something which is hard to imagine. But then to put it in a simple way, I've shown it like that. So you can see there are 3 grooves here. Those 3 grooves are meant for sodium ions to get attached over there. And since this is the potassium pump also, it's a sodium potassium pump, you can see I have drawn 2 triangular grooves where the potassium ions are going to attach. Now because of this pump, and there are other reasons also which we are going to dwell in future lectures, because of this sodium potassium pump, within the cell, I have mentioned here it's the intracellular fluid, and I have mentioned here as well it's the extracellular fluid. Within the cell, the concentration of potassium ions are more, and outside the cell, the concentration of sodium ions are more. Now I mentioned a very important word over here, concentration gradient. Now what is concentration gradient? If across the membrane there is high concentration of a certain thing, and on the other side of the membrane there's less concentration of that particular thing, then because of this difference in concentration, there is a gradient created, and this gradient is very important if you want things to move across the membrane, because it's a law of nature, or it's things pass from an area where they are more to the area where they are less, and this is known as diffusion. So if the things have to diffuse across the membrane, or they have to diffuse from one place to another, it is very necessary that the concentration of that particular substance should be high at one side and should be low at one side. And the movement of the substances are always from an area of high concentration to an area of low concentration. So do bear in mind concentration gradient. I'll be using this term also often in my lectures. And then let's see this sodium potassium pump. So you can see there are I've shown the sodium ions with black circles and the potassium ions in the blue triangles. So this particular pump picks up 3 sodium ions from within the cell. The 3 sodium ions get attached to this particular pump. So I show you 3 sodium ions attached over here. And once they are attached over there, ATP comes into play. So I'll show ATP since it's energy, high amount of energy. So let's do it like this. This is the ATP coming into picture. ATP, adenosine triphosphate. So the energy of ATP is used. The ATP is hydrolyzed into adenosine diphosphate, and one inorganic phosphate gets separated. That releases the separation or the breaking of the last bond of phosphate from the ATP, releases huge amount of energy that changes the configuration of this particular channel, and then this channel moves the opposite way. So using the energy of ATP, the pump turns around, and the 3 sodium ions which it had collected from within the cell, it pushes it or drives it outside the cell. Now these sodium ions are driven outside the cell. Now the same pump collects potassium ions, and of course, not to forget, the energy for this pump to work is got from ATP, the energy currency of the body, It collects now 2 potassium ions from outside the cell and then turns back again, turns around and pushes the 2 potassium ions which it has collected from outside the cell to inside the cell. Okay, so this particular pump then collects 2 potassium ions from outside the cell, turns around and pushes the 2 potassium ion inside the cell and comes back to its original position. So here are the 2 potassium ions in their binding sites. They have been collected from outside the cell, and once the pump turns around, these 2 potassium ions are pushed back from outside to inside the cell. So the pump is back to its original position, and this is the sodium potassium pump. It is working when the membrane of the neuron is resting, that is not conducting any impulse. Yes, it is using the energy of ATP to drive 3 sodium ions from within the cell to outside the cell and 2 potassium ions from outside the cell to within the cell. Now because of this, there is a concentration gradient created, and there are many potassium ions inside the cell of the neuron, and there are lots of sodium ions outside the cell. And this is the way the membrane of the neuron has a negative charge, because more positive ions going outside and less positive ions coming inside, only 2 potassium ions coming inside and 3 going outside. And this is also one of the reasons because of which the membrane of the neuron is negative inside and positive outside. The same membrane of the neuron has a membrane potential across it. That means it has a negative charge within its surface and it has a positive charge outside its surface. That's all about this pump.